Thank you for having me today and for scheduling this. And let me just dive right in. So, a fundamental aspect of biological organization that we think about is intercellular communication, which we know is necessary for the coordination and function of multicellular assemblies, and ultimately for the survival of organisms in ever-changing environments. So, in my research, I use the visual system as a model for understanding how different cell types work together for the formation, maintenance, and refinement of biological circuits at a few different levels of investigation. Um, okay, so during the development of an organism, we have neuronal stem cells, which proliferate and differentiate into neuronal and non-neuronal or glial cells. And then intercellular connections are set up during the initial formation of brain circuits, largely in response to genetic programs, for example, through molecular guidance cues that aid in pathfinding, navigation, and initial neuronal wiring. But for an organism to function optimally in its environment, its development must also respond to external cues. So, in other words, the organism and therefore the brain circuitry needs to have plasticity, which happens through refinement. So, the development and refinement of circuits confer a particular organization to the brain, which is thought to contribute to its function. And we see in many early sensory processing systems that they do have some sort of topography, right? So in the visual system, inputs from adjacent visual space are organized into adjacent neural space, which forms a retinotopic map. Similarly, in the auditory system, we have tonotopic maps. And then the olfactory system is a little bit different, where you have discrete maps for odorants. So, what are the underlying mechanisms that enable the sort of plasticity and precise refinement of circuits into fully organized systems? So, studies of brain circuits largely focus on neuronal differentiation and computation. But in the last few decades, we've seen that glia may contribute as well to this circuit formation and refinement. So to investigate neuron-glia interactions, particularly during activity-dependent circuit refinement, I turned to the Xanthus flavus tadpole. And most of the people in this lab know, but tadpoles develop externally from egg to adulthood. And that gives us a lot of access for genetic, pharmacological, and even environmental manipulations very early on in development. So this is briefly what their life cycle looks like, and a cool Tumblr GIF showing that life cycle. Um, and so we focus mainly on these mid-larval stages where tadpoles transition from relying on their yolk sac for nutrition to active feeding. And during this time, they demonstrate activity-dependent circuit refinement and more complex sensory motor behaviors in response to environmental cues. And the translucency of albino tadpoles makes them amenable to live in vivo 2-photon imaging, which is what I did. Um, so here is an example of a cell being imaged live. And we specifically studied the visual system, which is called a retinotactile system, where you have retinal ganglion cells, or RGCs, from the eye, projecting their axons to form the optic nerve, crossing at the chiasm, and then innervating the contralateral optic tectum, which is where visual input is first processed. And the tectum is thought to have a retinotopic map as well, which is this topographically organized representation of the tadpole's visual space. So importantly, we were able to study the effects of induced pattern neural activity on circuit refinement simply by controlling the animal's visual experience. So using flashes of LED light, this is a strobe stimulus, and it's been shown in the literature, depending on the frequency that you shine it at, you can actually synchronize all of the RGC firing. And that's a way sort of to control the underlying neural activity and then see how this affects things about that circuit. So most of the work I will present today is from my PhD dissertation, which sought to contextualize glial function and activity-dependent circuit refinement by investigating two distinct phenomena in the developing retinotactile circuit. First, with regards to myelin plasticity, we asked how sensory evoked patterns of neural activity alter early myelin unsheathment. And then second, we asked how a transmitter D-serine, whose availability at the synapse is modulated by both neurons and glia, alters the morphology and topographic map in the optic tectum. Okay, so let's jump into the first study, which I conducted in Ed's lab in collaboration with Dr. Thora Karadedir at Cambridge. So our understanding of circuit function tells us that the timing of neural activity is crucial, right? So this is, in your lab, you study this bioelectrical activity. So really, we're looking at how electrical activity is shaping circuit function. And this is in the nervous system. We know there's phenomena such as spike timing dependent plasticity that can do that. And then higher conduction velocity of axons allows information to be transmitted between neurons more efficiently. And so the solution to accelerate action potentials in vertebrate evolution has been to insulate axons in myelin, which is a lipid-rich insulatory material. So to demonstrate how myelination affects circuit timing, here's a schematic from a review showing presynaptic inputs arriving at the postsynaptic dendrite. So these inputs are able to summate and reach threshold in this bottom panel when there's myelination on board, which is indicated in yellow, because certain inputs, such as those with these longer axons, can arrive at the same time as other inputs. And so you get with myelination, it's not just a matter of amping up neuronal activity to the max, but it's a matter of how can you time the circuit so that you have better coordination between different regions. Uh, so this is why we think that myelination is important. And so in the past, it was thought that myelin is very passive. It just insulates axons. But now we have a lot of studies to show this other relationship also exists. So using tools like optogenetics, pharmacology, and electrical stimulation, it has been shown that neuronal activity affects myelination and can affect the differentiation and proliferation of the oligodendroid glia that are responsible for producing myelin. So all of these interactions in turn have been shown to then affect the circuit function of learning and memory and so forth. So given this interplay, my work sought to examine what are the links between myelination, neural activity, and then sensory experience, which is causing this neural activity in the system. So to study early myelin unsheathment, we have to start from the basics. First, we have to find an antibody that works for myelin, in this case, a specific protein called myelin basic protein, which is one of the most abundant proteins in myelin. And after a long adventure to find the right antibody. We found one. This is a Western blot showing it for rat monoclonal antibody for MBP, which was used on Xenopus adult brain lyset. And you see two bands corresponding to 19 and 22 kilodalton, which corresponds to different Xenopus MBP isoforms. And then you see the MBP immunofluorescence here in the adult optic tectum, as well as in the hindbrain of a tadpole at stage 49. And we observe this expected morphology of myelinated fibers. And also in this and all the subsequent figures I'll show, the sections are all horizontally sectioned, and they're oriented with the rostral to the top of the panel. So we cross-validated this MBP immunostaining using a label-free approach called third harmonic generation microscopy. And this has been used to image the presence of myelin in the nervous system. So I worked with Sarah Offencock, a postdoc, and Dave McFarquhar, a physics master's student, in our collaborator Paul Wiseman's lab to obtain these third harmonic generation images. So TG is a nonlinear optical process, and what it does is it's induced by the presence of an optical interface. So you can use it to look at these submicrometer heterogeneities in a refractory index. So in compact myelin, because it's compacted, there is transitions between lipid and the cytoplasm. You can actually use just that TG to differentiate it from surrounding neural tissue, which shouldn't have this lipid-rich content. So here's these TG images. And so again, this is the MBP panel. This is the TG signal. And you can see that there is basically where you see MBP is also where you see TG. And for those who have a keen eye, they'll notice that the TG signal, basically you see it in later stages than the MBP signal. And we think that's because, so MBP expression sort of starts early on, and then as MBP kind of gathers in more areas, it's also as myelin is condensing and gathering. And so you need a much more lipid content for TG to catch it. So we think that's why the TG signal is a bit delayed than the MBP expression. And because MBP expression is highly specific and precedes the onset of very compact myelination, we actually ended up using MBP immunostaining for the rest of the study because we were trying to look at this really early myelin enchantment and how that can be regulated by neuronal activity. So to investigate the effects of visual stimulation on myelin enchantment, we needed to know exactly where and when to look for these subtle changes. So I carried out a detailed characterization of myelin enchantment first, just during regular development. And for brevity, today I will just show you a snippet of that characterization. So this is the optic chiasm where the subsequent experiment was performed. So in yellow is 3A10, and then which is an antibody that labels retinal ganglion cell axons. And you're looking at the optic chiasm here. And then in magenta is MBP. And I first saw MBP, any MBP expression in the tadpole at stage 48. And then zooming out, this is just sort of an overview of the tadpole brain. You can see the changes in the brain across the course of tadpole development. So this is stage 48 and this is stage 51. And there's, barely any magenta that's seen. There's a bit in the hindbrain, very little bit in the optic chiasm. But then by stage 51, there's a lot more MBP expression as the animal is older. So based on our characterization of the time course and the location of early myelin enchantment, we focused on the effects of visual experience on MBP expression at stage 48, specifically in the optic chiasm. So as I mentioned before, we used stroboscopic visual stimulation at different frequencies to physiologically induce temporally pattern activity in the retinotactile system. And to characterize the responses of the RGC axons to strobe, we actually do 2-photon imaging of RGC axon terminals in the optic tectum. So these axon terminals are labeled by injecting purified mRNA for GCAM6 and mCherry into one blastomere of 2 cell stage embryos, resulting in tadpoles with mosaic fluorescent protein expression restricted to just one lateral half of the animal. And this technique was pioneered in the lab by Anne Scholl, who is a technician. Since tadpole RGC axons cross over to the opposite tectum, RGC axons originating from the label side of the animal innervate that tectum on the unlabeled side. So Vanessa Lee, a PhD candidate in the lab, recorded calcium activity in these RGC exon terminals while presenting animals with strobe of varying frequencies. So we piloted actually a range of frequencies, and I won't show all of the data, but the 2 important frequencies we observed, first of all, is this frequency, 0.0625 hertz, where you can robustly evoke calcium responses. So you'll see like a bunch of calcium transients and then off, and then a bunch of calcium transients, then off again. And then the other one that was interesting to us is 1 hertz, which is our fast strobe. And this one, basically, it's fast enough that it's only generating weak responses, and you're not seeing these like coordinated trains of activation. And you can see these movies as calcium traces, which is a little easier. And you can see for the slow strobe right
Can I
after
ask you a quick question? Sorry. You mind
yeah,
if
go
I?
ahead. Yeah.
So what are the on and off kinetics of the calcium of GCAP, the calcium sensor that you're using?
Yeah, so this is GCAM6. I can't remember the exact numbers, and
Mhm.
that's a really good point. And you'll see that in the fast strobe, you basically, again, I can't remember the
Yeah,
numbers
do you happen to know if
but
if it's yeah, I can never remember, but if it's the F, S, or M variant?
it's S, it's GCAM-S.
Okay, cool.
Yeah.
Yeah, so that one's I think it's like a second and a half or 0.9 seconds, something like that. Okay, nice.
Yeah, something like that. And that is one of the things we were concerned about because as if you just, if you just use something like a fast strobe, you might think that, oh, maybe it doesn't have time to wind down. But that's why we tested a range of frequencies to basically make sure that you're actually seeing it's not just an effect of the calcium kind of runoff kind of thing. Um, Right, so you can see that when the LED is flashed in the slow strobe condition, you have this peak of calcium responses, but not so much in the fast strobe. And these are just power spectra to show that the power peaks actually correspond to the frequencies, which is 0.0625 hertz here, and then at 1 hertz you have a frequency, you have a peak here. And then in the bottom panel, we're just showing that when you compare the stimulus frequencies, you have more power associated with the slow strobe than the fast strobe. And so we ended up using just these two conditions to assess the effects of different sensory stimuli on early myelination. So I raised animals under either ambient light or the slow or fast strobe for 7 days. So they just are, viewing these flashing lights for 7 days straight. And then I fixed animals and sectioned the optic chiasm. So in this video, you can see all the cell bodies labeled by the DAPI in blue, axons with 310 in green, and then MBP in red. And of course, where they're co-localizing, it looks yellow. And so for each stack, sorry, I'm just going to play that so you can see what it looks like. So I took the image stacks of each section containing the optic chiasm, export them to Imaris for 3D reconstruction of the chiasm and analysis in a blinded fashion. And so for each stack, the surface creation function is used for the MBP channel. So here's the MBP, surface creation, and you get that. And then, oops, okay. And then to study specifically the unsheathed axons of the chiasm, the 310 axon channel was masked with the MBP myelin surface. So now the information I'm getting at the end is specifically how much volume of axon is associated with MBP. And that's what we're interested in quantifying. So these panels demonstrate the representative snapshots of the reconstructions for the ambient, slow, and then fast conditions, just like what you saw in the movie. And then when I quantified how much of the axon is associated with MBP, you can see that our data show that slow strobe, which reliably evoked robust non-adapting calcium responses in RGC exons, increased the axonal volume associated with MBP, whereas fast strobe, which was associated with these weak adapting calcium responses, had a reduction in end-sheath axonal volume at the chiasm. So really suggesting that basically if you amp up the neuronal activity, you're actually also getting more of this very early myelin ensheathment, and vice versa if you're kind of just not getting these robust neuronal activity signals. So when it comes to early myelin ensheathment in the developing retinal detector system, our study provided a detailed characterization of what that myelination looks like, and that allowed us to go in at the right time and see if you could manipulate myelin ensheathment just based on the basis of neuronal activity. So in the second study, I'll talk about neuronal interactions in the same system, but through a very different lens. So here we're looking at the effects of D-serine, which is an agonist of the NMDA receptor, and how that mediates plasticity in the circuit. And importantly, D-serine is controlled by both neuron and glia. How much it's available at the synapse is controlled by both of those cell types. So just as a little bit of a background, the N-methyl D-aspartate type glutamate receptor NMDAR is thought to have a pretty evolutionary conserved role in the activity-dependent selection of inputs for circuit refinement. So NMDARs are heterogeneous in their composition, but almost all of them require simultaneous ligand binding of glutamate and a coagonist, which is either glycine or D-serine, alongside sufficient depolarization to actually relieve this magnesium block from the ion channel pore. And this concurrent ligand binding and membrane depolarization requirement for channel conductance is what makes these NMDRs ideal to detect temporal correlations of convergent inputs. So based on loss of function experiments in the literature, as well as a previous study from our lab, it seems that NMDA receptor activation contributes to a bunch of downstream signaling pathways, and that helps circuits mature. So going from, in the Xenopus, you have a lot of these exploratory growth when the cells are young, and then as the circuit matures, they actually stabilize morphologically, and that can affect how well also the retinotopic map forms as a result of activity. So we wanted to examine the postsynaptic effects of D-serine administration, which should enhance NMDA receptor activity and lead to hyperstabilization of the circuit. And the role of NMDA receptors in developmental plasticity, largely so far in the literature, is characterized through lots of loss of function manipulations. And that might actually disrupt normal network activity by reducing the overall neuronal excitation. So this is the same issue with, if you just throw on a bunch of glycine, which is the other coagonist, because we know glycine is an inhibitory neurotransmitter. So we wanted to use D-serine specifically because it would enhance existing NMDA receptor currents while still preserving this requirement for glutamate release. We didn't want to interfere with its ability to detect convergent inputs, but we still wanted to see what happens in sort of this gain of function when you use D-serine administration to push NMDAR activity to the max and see what happens to the circuit in that situation. So to visualize dendritic arbor morphology of neurons in the optic tectum, I electroporated tactile cells to sparsely express GFP, and then I performed daily in vivo 2-photon imaging. So after collecting the baseline of individually labeled tactile neurons, then I reared animals in 100 macromolar of D-serine, which we've shown in a previous study is a range that's not cytotoxic. It's and does have some axonal effects as well that I won't be talking about today. But yeah, it's a range that has been tested previously. So over the following 3 days, I imaged every 24 hours, and we saw that for neurons that are initially less mature, so when they have dendritic arbors of less than 500 microns, um, there is a significant reduction in dendritic arbor length. But that's not true for neurons that are already pretty large. Even if you remove this outlier, it doesn't really, D-serine doesn't seem to have an effect, which suggested that D-serine can change morphology sort of in immature cell types. And then for the dendritic branch tip number, similarly, there's a bit more of a difference between the D-serine and the control condition when the cells are immature. And then because we saw this difference, we sort of focused the rest of our analyses on these immature neurons. And we were able to do that by developing a modified electroporation technique that we published called Crimsicle. So that lets us target these really basically use Cre-dependent electroporations to target just these younger cells. And so next I performed Scholl analyses of these immature neurons. And as a reminder, in this analysis, you simply plot the number of intersections of the dendrites at increasing radii from the cell body. And so Scholl analysis revealed that over the 3 days, the D-serine arbors note how their branches stay closer to the soma. So really, all of this data together suggesting that immature neurons exposed to D-serine develop to be more compacted than controls. And we hypothesized that these morphological differences could have resulted from some sort of premature stabilization of the dendritic arbors of immature neurons in D-serine. So to test this idea, I investigated the branch dynamics with short interval imaging. So this is imaging every 10 minutes for 1 hour in animals that were exposed to visual stimulation in the form of stroboscopic flashes. So as daily imaging revealed a significant impact on by 48 hours, like by 2 days, that's when I started rearing animals in D-serine and then imaging the single cells. And then when I quantified them using a MATLAB plugin called Dynamo and some custom code I wrote, this is what the quantification tells us. So branches, which are any dendritic process that is more than 10 microns in length, exhibited a trend towards elongating less and showed significantly less retraction over the hour of imaging in D-serine reared animals. So overall, D-serine exposure significantly reduced the branch motility per time point over the hour of imaging. And so since dendritic branch morphogenesis was affected by D-serine rearing, of course, next we wanted to see if the synapses were also affected. And so to do that, I performed immunohistochemical labeling of presynaptic SV2, which is shown in yellow, and postsynaptic GLUA1, which is in magenta. And then basically perform quantification when both of these areas overlapped in ImageJ. And this is obviously a pretty crude method for estimating synaptic density, but we've used it previously, and it's a quick and dirty way to look at, if there are changes in synaptic density. And so, of course, we did see, I wouldn't be presenting this data if we didn't see that there is indeed a difference. So anatomical synaptic density in D-serine animals was significantly increased in the tectal neuropil. And then Anne and I also quantified the synaptic density of individual tactile dendrites. And to do that, we electroporated tactile cells, again at an early stage, to express the synaptic marker postsynaptic density protein 95 or PSD95 fused to GFP, as well as with DS-red, which fills the neurons. So you have the axon filled with DS-red, and then you have punctate of PSD 95 GFP. And when you quantify that punctum density, um, animals that were reared in D-serine for 48 hours had significantly greater number of synaptic punctate per dendritic volume. And then to exclude the possibility that D-serine may be acting, via some sort of non-NMDA receptor target, such as synaptogenic, like glutamate delta receptors or something like this, we reared some animals in D-serine plus MK801, which is an NMDA receptor channel blocker. And the addition of MK801 actually prevented this increase in synaptic density, indicating to us that the observed effect of D-serine is NMDA receptor dependent here. And then finally, to investigate how DC rearing may alter development of the retinotopic map, we turn to animals with GCAM6, again expressed in one side of the animal, as I showed in the previous study. And we reared these tadpoles in D-serine from stage 37 and image at stage 48 to cover the entire developmental period during which RGC exons first arrived to innervate the tectum. And so Vanessa recorded and analyzed calcium responses of the RGC terminals in the optic tectum while presenting receptive field mapping stimuli. So here, the way she showed receptive field stimuli for this experiment is something in one of 5 positions in either the azimuth or the elevation. So azimuth being like a horizontal and elevation being like an up and down direction to the tadpole. And so here's some representative examples. So these are the calcium traces, and then individual cell stomata were segmented from these 2-photon images, and these were used to estimate the receptive field center for each tactile cell body based on the relative strength of the responses to stimuli in each of these 5 positions. So here I'm showing representative responses in the elevation axes for control and D-serine conditions. And the maps are color-coded so by the optimal stimulus position that evokes a response in each cell. So if a cell is better at responding to a stimulus at the bottom of the tadpole screen, that cell is in blue. If it's better at responding to something at the top of the screen, then that's coded in red. And you can see there's a bit of a topographic organization here in the control and D-serine animals. But they do vary a lot from animal to animal. So to calculate the receptive field sharpness, the average fluorescence change for stimuli presented in the optimal position was divided by the mean responses to the stimulation of the remaining non-optimal stimulus positions. So a high receptive field sharpness value suggests basically a more compact receptive field. So here are the receptive field sharpness values mapped out to the tactile cell bodies. And this is the range here. And it's for me anyway, easier to look at it as a cumulative probability distribution. So in these light colored lines are each of the animals that Vanessa looked at. I think she had 6 D-serine and 5 control animals. And the bolded lines are the group wide, if the cells are pooled across the 2 groups. And so the D-serine condition actually shows a significant rightward shift, particularly in the elevation axis. So our data sort of suggests that D-serine rearing might lead to more compacted or sharper receptive fields compared to control. And this is sort of in line with earlier what we see with the arbor compaction and increased synaptic density for tactile cell dendrites. So it's as if when you have D-serine on board, you're hyperstabilizing and compacting everything or at least the dendritic side. So in this study, we showed that an NMDA receptor enhancement with D-serine administration led to morphological stabilization of the dendritic arbors and higher retinotactile synaptic density. And finally, this increased receptive field sharpness in tactile cell retinotopic maps. And one other thing that this body of work does is apply some new methodologies to, visualize things such as myelination, with TG, RGC exon terminals specifically by using these half GCAMP animals, and then also trying to specifically target recently differentiating neurons. So the work I presented thus far contributes to our understanding of the intricate and dynamic cellular interactions that occur within complex and highly adaptable brain circuits. But ultimately, why we care about this is to understand how that enables the circuit's function of processing information. And that's sort of what, brings me here today to talk to your lab, which is this interest in how cells are communicating to process information. So in the D-serine project, I showed you cellular changes may impact this visual retinotopic map. And the presence of maps, especially in sensory cortices, is a common motif, as I was saying earlier, for circuit organization across modalities and species. And neural maps encode a wide range of behaviorally relevant features into neural space. But another motif of circuit organization is also hierarchical processing. So in brains, neurons conduct information upstream, such as in this ventral visual system. And then as the information is conducted upstream, it's thought that now we're processing increasingly complex features, right? So from basic to really these conjunct, you have feature binding, if these complex objects emerge. So the existence of these sorts of motifs of organization of biological circuits, as well as all the metabolic resources that organisms have to devote to their formation and developing circuits, hints at the importance of organization for brain function. And while we know that these things exist, what hierarchically organized maps do in a functional or computational sense is not well understood. And the extent of her gap of knowledge in this regard really struck me when I was working on this review with Vanessa and Ed. But I'd already wrapped up my PhD at the time and was working in scientific communications, but I still was interested in this question. So I decided to pursue this basically as a hobby project in collaboration with Dr. Arnaud Gauche in the lab of Dr. Blake Richards at the Mila Quebec AI Institute. And we're sort of working on this story for publication, and it's the first time, I'm presenting it publicly, so please be kind. And you'll see it's really sort of outside of my domain as a biologist, but it was still very interesting to play with the data and see what we could get. So to answer the question of how these motifs of organizations might aid computation, we turn to a population geometry approach, and that's what Arnaud used in his PhD to characterize artificial neural networks, inspired by the work of Stringer and colleagues who are now at Janelia. So Stringer and colleagues showed mice 2,800 unique stimuli and recorded from about 10,000 neurons. So they obtained a matrix of n by n neuronal representations. And since they had at least two presentations of each stimulus, they could perform cross-validated principal component analysis, or CVPCA, which measures the reliable variance of stimulus-related dimensions. So in other words, you can use CVPCA to exclude trial-to-trial variability from unrelated variables or noise, and allows you to look at the variance that is specifically related to the stimulus that you present. So next, they plotted the eigenvalues of the cross-correlation matrix, observing an almost linear slope of decay in a log-log plot. So the eigenspectrum decay for these mice, V1 representations, it turns out can be characterized as a power law of the eigenspectrum decay. And in mice V1, that's approximately 1. So now we wanted to use the same approach of taking this cross-correlation matrix and plotting out the eigenvalues and then seeing this decay, because that can be used to look at representations in any network. And before I sort of present our preliminary results, I wanted to take a second to explain how that power law fit has anything to do with population geometry. So to illustrate the consequences of eigenspectrum decay, Stringer and colleagues simulated neuronal population code responses to a one-dimensional stimulus in the x-axis, and then they visualized them by random projection into 3D space. So a low-dimensional code with two non-zero eigenvalues, right? Two non-zero eigenvalues, that produces a circular manifold. An uncorrelated high-dimensional code where each neuron responds to a different stimulus produces 1,000 equal variances. And this is sort of what the efficient coding hypothesis is, right? Like one neuron is only coding for one feature, for example. But if you do that, you get a projection where it does not respect distances. So things that are should be encoded similarly to each other are as encoded as differently as things that should not be encoded as similarly as each other. So you get this spiky discontinuous ball. But what's interesting to us is these power law codes, which show a scale-free geometry. So these codes are much smoother. And what happens here is a power law code with alpha is equal to 2 is non-differentiable fractal because the many dimensions that are encoding fine stimulus details, which are in blue, outweigh those that are encoding large stimulus differences, which are in red. So the neural manifold here is on the border of differentiability. So all this means to me as a biologist is when you have a structure like this, you're able to preserve these really fine scale features as well as coarse features in the representations that you're interested in. And then recall that Stringer and colleagues found that the power law fit in mouse B1 was approximately 1. So it turns out that for that setting, because this is a one-dimensional setting with just the x-axis, right? But in a biological setting, what they found is alpha of 1 was a value at which the eigenspectrum of the visual cortical code can decay as slowly as possible, such that both these coarse features and fine features are preserved and remain differentiable. So to investigate whether scale-free representations are universal, we characterize the eigenspectrum of the activity covariance of individual brain regions in the visual cortical hierarchy, and we use the natural scenes data set. So the natural scenes data set is a large-scale 7 Tesla fMRI data set consisting of whole brain high-resolution fMRI images of 8 healthy subjects while they're viewing thousands of color natural scenes. So R9, I use these activity responses, and we use the basically parcellation, well-known parcellation, to plot the eigenspectrum of early, intermediate, and higher order areas. And so these are just some representations from one subject. And you can see that the power law fit for the early and intermediate areas is pretty good, but in these higher areas, it doesn't really seem to be a power law decay. And that's what we quantify here. So here's a power law fit that we're quantifying using the R-square coefficient. And we see in early to intermediate areas, there's a pretty good fit of about 0.8. But in those later areas, it's really a bad fit. And um, this to us really indicated that early and intermediate areas have the scale-free geometry, the one that can preserve both coarse and fine features, but higher order areas do not. And when we look at what the value of alpha actually is in V1, we see similar to what Stringer and colleagues show and what people show in monkey and humans. For V1, indeed, alpha is 1. And when we look at the rest of this cortical hierarchy, alpha remains pretty close to 1, all the way up to intermediate areas where there's a good power law fit. And then it just, again, the alpha no longer holds. So as a control, we also looked at a different visual hierarchy that should not be involved in this task, which is the dorsal visual hierarchy. And the dorsal stream is responsible for motion detection and not visual recognition. So this stream really should not be recruited during the NSD task. And we see that indeed there's not much changes in alpha for the dorsal stream. So altogether, that tells us that looking at the ventral visual hierarchy in early and intermediate areas, you see a scale-free representations, but these are not universal and largely are not present in these higher order areas, at least for this data set that we were looking at. So why do we care that the power law changes across this hierarchy? So we think that when you have scale-free representations characterized by a power law decay in the eigenspectrum, that allows early and intermediate layers to learn different features at varying scale, and that makes it robust to certain noise statistics in the inputs. This means that these representations are generic enough to support a large variety of downstream behavior. And why they may not be needed for higher order regions is because these regions are actually functionally specialized and they care about supporting only a few related behaviors, uh, which is what the higher order visual areas are doing, right? They're doing few shot learning. And so they just need uh, they don't need to preserve all these fine features. They might only need to work with coarse features, for example. So our findings lead us to postulate that hierarchical information processing allows early and intermediate areas to solve these robust representation learning and then provide generic enough representations that can be used by functionally specialized areas to then make very task-specific responses. And now we're hoping to test this relationship between the power law exponent and generalization versus specialization in an ANN setting, because obviously it's very difficult to do that in a biological setting. So in summary, my work explored neuron-glia interactions in the context of a developing sensory circuit, showing how experience can change the form and function of biological circuits. And then I showed you some unpublished data using population geometry approaches in the visual system to understand the links between organizational motifs like hierarchical organization and the function, which is computation in brain circuits. And so together, this body of work contributes to an understanding of biological circuits through different lenses, and I hope to continue pursuing that through my future research projects. And before I conclude, I'd like to thank the countless people who contributed to this body of work in big ways and small. And I already tried to highlight the contributions of my co-authors throughout the presentation, but I will also like to thank the rest of Ed's lab, advisors and collaborators from around the world, and our funding sources, which were all necessary for these projects to take place. And I'll take any questions.
Excellent. Thanks so much. Question.
Yeah, I'll start off if you don't mind.
Yeah,
So
sure.
I think it's really cool how you're showing that circuits can be changed by the extracellular matrix essentially, right? Because the myelin, and so we study non-neural bioelectricity, and one thing that we're often trying to do is translate things that we see in neuroscience into non-neural systems and see how in this more primitive system they behave. So I guess do you think that in non-neural systems, other extracellular matrices, because the stem cells don't have myelin, do you think that other ECMs could be acting like myelin and sort of shaping the non-neural bioelectricity?
Yeah, so I think there's definitely, I think, an interaction between, I think electricity can shape things around it, and then things around it can sort of feedback and shape electricity. I think that's possible. I don't know to the extent of what they would act like a myelin. If we're talking about myelin sort of being, a lipid layer, um, perhaps there's in nature, even animals that don't myelinate have myelin-like substances. So definitely, I think there might be other things going on. For example, sugar-rich molecules, right, that you could also use that would perhaps change how you're conducting electricity. So I think there's definitely a lot of room for crosstalk, and a lot of it is largely unexplored. And in this lab, you probably talk about it a lot, and it seems common knowledge, but I think outside of certain niches, most people are not looking at those questions or are not, even the idea that this neural activity can change myelin directly is sort of controversial, despite the evidence in the last 10, 15 years to really show it. So yeah, I don't know if that fully answers your questions,
No,
but
that
yeah.
that's great. Yeah, thanks.
Any other questions?
Anybody else?
Sorry, my mic was muted. I'm really good at Zoom after all this time. Uh, can you send, or I guess, Mike, do you have a list of some of the papers that Zara referenced today, just so that I can look at them like on my own?
Yeah, Zara, do you wanna send out some is a
Yeah,
list?
I'm happy to. Is
Oh, that'd
there
be awesome.
Yeah. The last one is not, as I said, published, but I'm happy to send like the Stringer's paper, for example, if that's something that's interesting to you.
Cool, cool.
Super.
Can I ask just a sort of a technical? How do you keep the frogs still
How
when
do
you're
we
doing
keep
yeah, when you're doing like live imaging? Uh,
Oh yeah, we actually embed them in a custom-made chamber that's like
oh, cool.
yeah, it looks kind of like agar, but yeah, you can also anesthetize them, but we don't do that for the most part, especially for the visual stimulation experiments. We just hold them really tight, like the chambers. Basically, we have one for different stages, that somebody had to make at some point in the lab.
Yeah,
Yeah,
and so I mean, do you fill it with agarose or something? Just because, like with
yeah.
the two photon stuff, I mean, you can't have them move like a micrometer and they stay
Yeah,
so still.
so
So
actually the one that Vanessa uses is much more custom made. It's sort of like built with the LED screen on the side, and yeah, it's kind of an agar. I don't think it's agar, but it's a related material. And it's basically pretty much translucent. She keeps it in the chamber, and also the screens have to be really close to the tadpoles for them to be able to see the entire elevation and azimuth axis.
cool.
Yeah.
Thanks.
I got a question just to follow up on that. There's been arguments over the years as to whether the tricaine that we're using on our tadpoles is actually an anesthetic or just a paralytic mostly. And so my
Mm.
question is this: have you found out whether once you, let's say, tricaine them and you show them images or just light exposure in general, do you see what do you know? Does it actually remove the signal that you're trying to track, or is it still there?
We usually still do see signal. Yeah, it's something we worry about too. Like we're able to see calcium signals, but we still wonder if we haven't systematically been like the other thing we use in the lab is pancuronium for some of the kind of imaging that we do.
That
Um,
is, that is, if I'm not wrong, that is just a paralytic, right? That's a
yeah,
postsynaptic model. Yeah.
yes, exactly. So we haven't done a systematic comparison, but my guess is it does do something. But we're using it equally in the control and experimental group. So we're hoping whatever it does, isn't a confound to what we're doing. But yeah, I totally see your concern. Yeah.
Yeah, well, right. No, it's more of a bigger issue. I wasn't worried about your data specifically. It's like one of the things that I'm thinking about is that this could potentially create a model system for a really interesting phenomenon, which is that in humans under what looks to be general anesthesia, and so you're in an OR, the anesthetist will say that you're completely out and all of that, and then some percentage of the people will report experiences or like literally specific. Oh, I remember when the doctor dropped this thing and everybody was talking about it, whatever. Like there seems to be some, and there's a whole field of basically subconscious learning, showing people subliminal messages that they have no overt recollection of, but that it somehow impact behavioral assays later on and so on. I wonder if we could have some sort of a minimal model here where you could anesthetize them to various degrees and then recover either just computationally or behaviorally. Ask what exactly do they still what experiences did they actually have, despite the fact that certain parts of the brain might have been shut off.
Yeah, yeah, because yeah, you could just read out their calcium responses and try to ascertain from there, and then try different types of anesthesia. Yeah, I think that would be interesting to see. And I think the problem with anesthetics is a lot of the times, at least the ones used in human, the entire mechanism is not known, and they're still quite widely used in medical settings. So I think there's lots of unexplored questions there of
Yeah,
downstream.
but in your case, you would like we would actually have, by imaging the brain, we would actually know which regions are active and which are not, which might be.
Absolutely, yeah.
And could you use the the myelin ensheathment plasticity to basically see if they're learning? To say that even when these things are knocked out, their myelin's changing, so they're learning.
Uh, that would be a stretch in this system because one, you'd have to show learning, and then you'd have to show changes. And I'm showing
Well,
you
I guess
really,
hysteresis, not necessarily learning in the neuroscience sense, but just seeing the changes in the myelin patterns as a result of
yeah.
the
So at the very early stages of myelin enchiethment, these are just aggregations of MBP. So it's not necessarily, we don't know how much myelin is there. We don't know if it's enough to have any change in neuronal conductance yet, right? And then actually really good myelination is a much later event in Xenopus. So what I would say my PhD shows is Xenopus is not that great of a model for early myelination, but a great model for like adult myelination because of course you have regeneration and that sort of thing. And I think that's a good place where you could test learning and myelination and demyelination and people look at demyelinating effects in adult frogs. Yeah.
I think it'd be interesting as well. Have you seen there was a guy that used that stringer paper to do some analysis. His name is Woodrow Shu. He's at Arkansas, looking at power law dynamics in those recordings. These were ePhys recordings, so not well, no, they were calcium recordings. Oh shoot, no, I don't know.
They're probably calcium
I
because
think they're calcium because it's stringer, so that would make sense. But if you look at the entire population, it's not power law at all. There's like subgroups of the population that are like have these power law dynamics, not the whole thing. And what was really cool is the ones that were like the subpopulations that showed power law dynamics that have these kind of scale-free representations were often most associated or kind of paired or correlated with scale-free behaviors like whisking or eye movement. So it may be cool to do something very similar in the data that you have, because if you are doing imaging in, another organism, if that property holds, which I assume it would, that would be super cool.
yeah, so that's really interesting. I didn't know about that paper. And if you don't mind like writing his name in the chat, I would love to look
Oh
it
yeah,
up.
sure,
But,
sure, sure.
I think, yeah, what was interesting to us, because generally with power law and scale-free geometry, people, have a tendency to think it is universal. And so we were actually surprised to see that in these higher order regions, you really don't see these power law fits. And despite this data set is an open source data set, it's existed for a few years now, there hasn't been papers yet looking in these higher order regions in humans. So I think
Hmm.
that was really exciting for us. And
Yeah,
I completely agree that power law is probably not a universal property. But it makes a lot of sense for when you're trying to have a lot of features that your system is just trying to hold on to use for things down the line that are more specialized. So yeah,
cool. Thank you.
thank you.